Welcome everybody. This is Francisco Rodriguez with Supplement Innovator. I'm excited to start today's webinar. It's going to be very insightful. We're going to be focusing on probiotics, and we are going to get started in about 3 minutes. Thank you all for joining us, and we look forward to getting your feedback. If you have questions, feel free to email me at francisco@supplementinnovator.com. And if you want to post your questions on the YouTube page, you can go there, post a comment, and we'll be responding to those. I know that many of you that are on don't necessarily have probiotics in your product line. Maybe you're a raw material supplier that supplies a number of different ingredients. We're going to be talking about that next week, but I think you'll get a lot of insight from what we're going to talk about this week with regard to probiotics and the microbiome. So let's go ahead and get started. I'm sorry for being just a minute late. Welcome everybody. This is Francisco Rodriguez with Supplement Innovator. Supplement Innovator is a consulting firm that supports ingredient sales teams and finished product brands, and we support those two levels of the value chain to create great products. We do sales training, and we do product formulations that are provided to the marketplace in the different market channels. We also do what we're doing here today, which is a focus on what's next for the industry, and we wanted to focus on the microbiome. And we brought in Take Ogawa from Second Genome. Second Genome is one of the most innovative companies in the space of next-generation sequencing. They have fantastic capabilities. They're working with major companies in the pharmaceutical and digestive health areas. They're also working with academics globally, and we invited them here to specifically talk about today the world of the microbiome and probiotics. And so you can learn about Take in some of our original videos, but I just wanted to start with the presentation and then open it up for questions. So without further ado, I want to open it up to Take to present to you the microbiome and probiotics. Take, welcome.
Thank you, Francisco. Appreciate the introduction. So let me get set up here while we set up the presentation. So let's see. I think I'm supposed to do this a different way. I apologize. Let me do this. I think I'm supposed to share my entire screen. So let me do that. And then do presenter mode. Great. So again, thank you, Francisco, and apologies for taking a couple seconds there to set up here. So again, what we'd like to discuss today and dive a little deeper is the idea around probiotics and the microbiome, and of course probiotics being live organisms themselves, and how they are at interplay with specifically the gut microbiome and how that relates to health outcomes, new product concepts, and so on. So we want to discuss that in a little more detail today. So again, a review for everybody that's joining the webinar here today. When we define the term microbiome, really what we're describing there are the various microbial communities that, of course, consist of bacteria, viruses, and yeast. But in this case, we're specifically focused on bacteria today, of course, when we talk about probiotics for the most part. And when we look at these microbial communities, these commensal microbial communities that take up residence in various parts of our bodies, it turns out, of course, that depending on where we're looking at, so which part of the human body we look at, that different members of those communities reside there. So different species, different strains of organisms reside in a very unique fashion across all human beings, depending on which body part we talk about. So this is just depicting the diversity that we see and how they're unique to different body parts. And particularly today we'll be focusing on the GI system here, and the GI system has one of the most complex and diverse microbial communities, microbiomes that is, that reside there, which have been directly associated with various functions that are that necessitate our well-being and our health status. And so here's just some description, high-level descriptions of how these microbial communities our interplay with our own biology specific to the gut. And I highlighted there in red specifically that it has been shown that these microbial communities have a direct impact on our metabolism, as well as immune response, inflammation, of course, and various other critical biological functions to our well-being. So what we, of course, want to understand is how do we perturb or affect these microbial communities to restore balance or to enable continued balance of a healthy homeostasis, or to correct for dysbiosis, rather. So this is exactly what we're trying to understand today, or what we will be investigating further today, I should say. So to take that point home a little further, there is this idea that as our microbiome is in imbalance, that there are certain outcomes in our health status or clinical outcomes even that we see. And so here's just some examples of the effects of an imbalanced microbiome or a microbiome in dysbiosis. Some of the things we might see, such as increased cases of infections, diarrhea, various GI disorders, and long-term effects such as obesity and allergy have all been tied back to imbalances in our microbiome. So when we think about how we assess the microbiome, there's been a long history, of course, of how we have researched and studied microbial systems, microbial ecologies, and bacterial strains specifically. And you can see that we've been doing this for quite a bit of time, but as technology and science has evolved, we have shifted the paradigm from just looking at one microbe at a time in a traditional cell culture assay, for example, or looking at them under a microscope, for example, to now we have been able to exploit various technology advances in genomic assays, looking at the DNA, that is, of these microbes, and of course through various advances in computer science, where we've been able to take this large aggregate of data that is enabled by these DNA sequencing technologies, as an example, to elucidate and characterize and understand what we're seeing there at a community level, rather than just at a single strain level. And of course, that has enabled a total shift in the paradigm of how we look at probiotics and their effect in human health. And so, as I described again, rather than looking at single strains at a time, and keeping in mind when we talk about culture assays or looking at single strains at a time, is we can only look at what we can capture or culture. Now, through the advent of, again, genomics technologies and various advances in that arena, we don't need to culture them, number one, and thus we can do culture-independent assays. That is, that we can look at the entire bacterial community, regardless if they can be cultured or not, in a totally unbiased way. And so there's been a total paradigm shift in being able to, again, access the whole picture of what's happening in a microbial community. And so that allows us to ask different questions when we enable or start to conduct various clinical trials on the effects of, say, probiotics to our health, is rather than just looking at the outcome of, say, patients or subjects that are on a particular probiotic or treatment, that is that now we can start to tie the mechanism, or the function rather, of how these probiotic treatments are affecting our gut microbiome, and thus how are those changes in the gut microbiome tying to the outcome. So we're allowed to ask much more hypothesis-driven questions about the mechanism of these probiotics with these new changes in technology and insights that we've gained through those. And then also it allows us to ask the third bullet point there, which is what are the effects of the probiotics to the gut microbiome itself, such as that are these probiotic strains colonizing and taking over our gut microbiomes, or rather is it that there's a much more subtle effect there, that it's actually modulating the community that's already there and changing their relative abundance and changing the makeup of those communities in much more of a symbiotic way. So now with the change in how we look at the microbiome, we can ask these very pointed questions of how exactly are probiotics at play in our gut. So what that allows us to do, rather, is again, rather than just looking at one species at a time, so we'll use an analogy here of say we go to the Serengeti or some sort of ecosystem that has an analogous complexity as a microbial community does, is rather than just looking at one species, that is one animal at a time, we get to go beyond that, and now we're not talking about just a single or even two organisms, but rather we're looking at a very complex ecosystem in one shot. That is what is enabled by the microbiome research that is capable today, is that we can look at the entire ecosystem, the entire ecology of all the different members that make up that ecosystem, and of course how is it at interplay with the host system itself. And so that is what has been enabled by the advances that we've seen in the science and the technology. And all of this is accessible to everybody today, that this is not particularly a niche application or some novel technology, that through various collaborations and partnerships, all of these types of science and trials are very much available today. So the kinds of data that we can generate, this is just an example here where to highlight the kinds of results that we can enable through the microbiome research that's available today, is when you take an individual and sample their gut microbiome. So in this case, we were sampling their stool samples, which is a proxy of course, the microbiome of the gut, is that we can see that in this example, that treatments such as antibiotics have a very dramatic effect. And so as you go through this time course of an individual that's been on an antibiotics, you can see from daytime 0 that these different colors represent a different species of a bacteria, that the diversity, of course, is relatively high before treatment, but then once they go on to treatment, that basically the diversity is completely wiped out, and only after a year or so does that diversity start to recover. And this is just one example, one study that demonstrates this. There's been many other studies to show that there is such a dramatic effect with these basically universal antibiotics that were so commonly prescribed in the medical community today, not only in our country but in many other countries, of course, where again we're seeing such a dramatic effect in the gut microbiome, and now we can apply real science to measure exactly, to quantify exactly what those changes are and how quickly does our microbiome recover after this severe perturbation of our gut microbiome. So another study that's very similar to that is now a study that was published just a couple of years ago, but in this particular study they were looking at the effects directly of a probiotic. And in this case, each column represents a separate individual that underwent a particular treatment of a probiotic here. And you can see at a high level there, when you look at the bar chart at the very top there, this is just simply counting the number of separate taxa, that is, separate organisms that we were able to detect within a given individual. So you can see when you don't have an unbiased point of view of the complexity of a particular person's gut microbiome, is that we detect over 1,000 different organisms in a per individual basis, and of course it differs depending on individual to individual, but for the most part uniformly you can say that there's high complexity, high diversity in a given individual. Now, the probiotic that we're given was a particular strain of a lactobacillus, which is designated here as LGG. So if you focus on the second bar graph there, designated as B, now they're looking at the relative abundance of a particular strain of that probiotic that was supplemented in these individuals. And you can see there's tremendous variation here from individual to individual as to the relative abundance of that particular strain. So again, this goes to show that even if you give a group of individuals the same dosage of a particular strain, that the colonization of that particular strain can vary significantly. So we need to dig deeper than that, rather than just looking at colonization, but rather what is the effect of these colonies of a particular introduction of a probiotic on the overall diversity or overall makeup of a particular person's microbiome. So that's what they exactly did in this in this given study. Was that what we're looking at here is a hierarchical clustering. That is, we cluster the overall similarity of an individual's microbiome, and so the closer they are in a branch here, the more similar they are as an overall makeup. That is, the members that make up that microbiome. And you can see that even though the colonization of the LGG differed across individuals, that the overall makeup, the overall diversity, the overall signature, rather, of these individuals that had high levels of LGG clustered together, that their overall community were very similar to each other. So you can see that there's a symbiotic effect of individuals that were taking this probiotic, that there was a clustering of their overall microbiome that became very similar after this treatment. So this is just another way to look at the data that we do commonly. This is a common output that you would get when you do a microbiome study. So what does the microbiome bring to us, of course, is it allows us to decrease the fear that we have around around specific bacteria. Of course, bacteria has a bad name in this sense in the general community, but by able to looking at the overall microbial structure, that we can identify each organism accurately, we allow the community as a whole, the market as a whole, to start decreasing the fear of the bacteria, because now we can accurately measure exactly what's happening there. And then by able to quantify those total organisms, of course, it allows us to start to differentiate how each of these treatments, that is, each of our products and or probiotics, are differentiated based on this data, because now we can quantify the entire signature of what's happening in the gut. And then, of course, this allows us to think of new products, because now we can see overall what's happening at the microbial landscape, and by identifying and calculating those shifts based on these treatments, we can think of new ideas. So it's much more of a science-driven, hypothesis-driven way to run these trials, and thus use real data, real science, to start differentiating the way we do development, and as well as how we differentiate our products in the marketplace today. So in the end, what we're trying to do is not just identify the organisms that are at stake there within the gut, and how our products are changing that, how our probiotics might be changing that, but rather, in the end, from a scientific standpoint, we want to be able to characterize the function of those changes. So if we were to say that certain strains or certain organisms are more abundant because of a particular treatment or regimen, we want to understand what that means at the biological level. What does that mean to us as a host, and what does that mean to our health? And so when we start to characterize these organisms as a community, we can start to elucidate certain pathways and certain functions that are implied by those pathways, as depicted there in this blue section, which is one specific metabolic pathway. And there are, of course, many different pathways that are implicated by these bacterial communities. And those pathways then start to elucidate for us the metabolites that are generated by these bacteria, and various other beneficial bioactives, such as short-chain fatty acids, which have been shown to have very beneficial impact in our gut health and our overall health. And so we can start to connect the dots here in that sense of what's happening at the biology, and how does that tie into the shifts that we see in the bacterial communities at hand that we measured. And so that enables us to start to engage on another type of study that's depicted here. So this is a study that came out of Europe, such as groups, Nestle and various other groups were involved in this study. So in this particular study, they were looking at over 150 different participants, where it was a split between male and female participants, of course. And based on different treatments of a probiotic at different time points, samples were collected, in this case stool samples, and certain individuals were on a placebo treatment, and again stool samples were collected. And when we look at the effects of this this probiotic treatment in this clinical trial type of a study, we do notice that when we start to dig into specific organisms, that the male and the females separated quite significantly. So statistically significantly, we were shown that certain organisms were distinguished between the male participants and the female participants in this particular study, that is. And they were taking male and female that happened to be obese as well in this particular study. And so what does this all mean? And so what it boils down to in terms of the conclusions were that women on this probiotic were also shown to have relative weight loss compared to women that were not on this particular probiotic, and that within the microbiome, that a certain family of organism was shown to be correlated with this weight loss, in that the particular treatment seemed to have shifted this particular organism within the gut microbiome, and we were able to measure that, or rather this study was able to measure that. Second Genome was not part of this study, just to be upfront about that. And that this particular family of organisms, when we go into the literature, is shown to be associated with a regulation of obesity and type 2 diabetes. And so now we're starting to, again, connect the dots between a clinical outcome and the shifts and the changes that we're seeing in the microbiome. And I'll stop there and hand it back over to Francisco. And take any questions at this time if there are any.
Take, thank you so much for that presentation. I hope the people that were on really got to appreciate what we were seeing. I have two questions in particular, and what I want to know, I wrote them over on the YouTube channel, and so what I want to make sure that we're all clear on is, number one, is there evidence that probiotics support existing diversity, and how has that been typically measured if most people that were measuring gut ecology were using culturing? And so basically the importance of probiotics supporting existing diversity and how it's measured, and why is that important?
Right. So it has to start in the beginning, and what I mean by that is when you have a hypothesis of that type, where you want to know exactly how your particular probiotic product is changing the gut microbiome diversity or richness, that is that you have to, of course, it all comes down to your study design and the samples that you collect in that context. And so putting together an appropriate study design where you're collecting samples before treatment, during treatment, and also post-treatment to be able to measure accurately how the microbiome has been changing as it associates to the particular treatment at hand. That's a big part of it, is that you have to have those samples collected, and collected in a reproducible way that you can actually ask questions of that level. And then the second part of that, of course, is that you are actually employing the proper methodologies in terms of how do you accurately and reproducibly measure the microbiome diversity. And that's, again, what's been enabled by the advances in the technologies that I touched on earlier in terms of DNA sequencing and such, is that you want to be able to measure that gut microbiome diversity in a totally unbiased de novo way, that anything and everything that is there is accurately captured and measured. And that, of course, is enabled by genomics technology such as DNA sequencing, because we all know that bacteria have DNA. And so if the DNA is there, this technology will measure that and accurately identify which bacteria that DNA came from, as a very simplified depiction of what we're doing here. But that's the basic idea there, is that we're not only measuring what we can, but we're measuring anything and everything that might be there, and how are they changing based on the questions that you have, as you described.
So adding to that question, and I don't think it was clear because we were comparing culturing techniques to now what's called next generation sequencing techniques.
Mm-hmm.
The limitation obviously was, possibly a small group of bacteria, but typically one that you're isolating versus getting all of them. But I think there's one other one that maybe wasn't touched on in the presentation that I want to be clear, and you and I have talked about this. You can never get in culturing those bacteria inside the gut that are unable to be cultured. Can you talk a little bit about that?
Absolutely, and that is true. I neglected a very key point there about the differentiation between culture-based and culture-independent methodologies, is that in a culture-based approach, there's a big assumption there that, number one, you can isolate the particular strains of interest and then literally culture them in a laboratory environment. And as we know, bacteria are organisms in their own right, and they're very sensitive to the environment in which they will thrive and thus grow in a culture environment. And so it turns out, to put some numbers around it, it turns out that over 90% of organisms that we find, say, in the gut, cannot be cultured. That people have tried to culture these, and they have been so far unsuccessful in culturing them for various reasons. Some are anaerobic, some are aerobic. They require different media. They require, that is, they require a different environment to thrive, and a lot of these environments cannot be replicated outside of the gut. And thus, the only thing that's universal across all these complex organisms is that they all have DNA. And so that's the big paradigm shift here, is that regardless of if they could be cultured or not, because they all have DNA, we can analyze them using genomic technologies and thus identify even organisms that cannot grow in a culture-based technique. And so, as I said, there's a high number of those 1,000 different organisms that thrive within our gut. Good majority of them cannot be isolated and cultured, and that's exactly why we need these enabling technologies, such as you described, next generation sequencing, to start to characterize those and identify those, and that's what's happening today.
Well, that's fantastic. I'm glad that we touched on that last point, because I know that was an important distinction that we talked about, and most people don't understand that. I didn't realize that it was 90% of bacteria in the gut can't be cultured on a plate. And so most of the people on listening to the webinar right now, they're familiar with identifying bacteria on a plate and then selecting that bacteria to then genetically sequence it if they do 16S sequencing. Others might know that they just plate them and identify them by their morphology or the way that they look. The problem with both of those is that 90% of the gut bacteria cannot be cultured. I can't emphasize that point and overemphasize that point enough. I think that was really helpful. So when we consider those things, and I'll leave us with this, when we consider those different components of the ability to analyze the bacteria in our gut and the reality that we have probiotics that will support diversity versus antibiotics which should destroy diversity. And if you go back to the previous webinar and the introduction webinar, specifically, diversity is a measurement of health. And it is a measurement of health from the infant all the way to elderly people. And so if antibiotics are destroying diversity and probiotics demonstrate some supportive diversity, what other implications are there? And I think we saw there's some correlation to weight loss in obese populations, and potentially some other areas that are being constantly uncovered. So great webinar, great content. Next week we are going to be focusing on specifically the microbiome beyond probiotics. As we talked about in the world of nutrition and diet, we had some ideas of proteins and of course fibers make a dramatic impact on the gut flora, and their quantities matter and their types. So the type of proteins, the types of fibers, the types of foods, possibly polyphenols, vitamins. And so when you look at all of those possibilities, the world of the microbiome is not just another story for the probiotic sector of the dietary supplement industry, though I think it's an important one for the probiotic groups, for the probiotic brands and suppliers. But it also opens up for the rest of the industry, because we're talking about digestive health, which also affects the ability to absorb nutrients, extract energy, and potentially have downstream health benefits that we are still looking to move from correlation to causation. So next week, we're going to be joined again by Take Ogawa. Take, thank you again for joining us, and everybody that was on, and those that get to listen to the recording. We look forward to seeing you or you participating next week, and feel free to shoot us questions ahead of time. We'd love to hear from you, and thanks again for being a part of this webinar.